I want you to look across the room right now. Or, if you aren't at home, just think about the last time you were in your living room and picture a houseplant.
Yeah, just an ordinary houseplant.
Right. A fern, maybe a pothos or a ficus, sitting quietly in the corner. You look at it and it seems completely still.
It basically just looks like a piece of green furniture.
Exactly. A piece of green furniture. And you would be completely forgiven for thinking that absolutely nothing is going on inside of those leaves. But today, we're going to completely shatter that illusion.
We really are.
Welcome to the deep dive. We are thrilled you're here with us. And our mission today is to explore a truly groundbreaking piece of research that changes how we view the natural world.
It really fundamentally shifts the perspective.
It does. We're pulling our insights from a January 2026 paper published in the journal Nature Plants.
Yeah.
This was led by researchers Trupti Gaikwad, Murray Grant, and an incredible team of scientists. And, what this team has managed to do...
It feels like science fiction.
It really feels like it belongs in a science fiction novel. They have essentially decoded and literally visualized the invisible, high-speed electrical and chemical alarm systems that plants use to protect themselves from disease.
Right. And to really appreciate the magnitude of what this team accomplished, we need to lay down the foundational concept they were investigating.
Lay it on us.
It's a phenomenon known in biology as systemic acquired resistance, or SAR.
Okay, SAR.
Yeah. Imagine a plant detects a pathogen. In the context of this specific study, the researchers were working with a bacteria called Pseudomonas syringae.
Pseudomonas syringae. Got it.
When that bacteria invades a leaf, the plant doesn't just sit there. It doesn't passively let the infection spread through its tissue. It mounts a defense, and the first step of that defense is incredibly intense and highly localized.
How so?
Well, the plant essentially commands its own cells immediately surrounding the bacterial invader to just die.
Wow. A scorched earth tactic, basically.
That is the perfect way to describe it. A scorched earth tactic. It's scientifically referred to as the hypersensitive response, or HR.
HR, okay.
The plant willingly sacrifices a tiny microscopic piece of its own leaf to trap, dehydrate, and essentially starve the bacteria.
That is intense.
But that localized sacrifice is really only half of the story, and it's actually the less interesting half.
The less interesting half.
Yeah, because the far more important function of that infected area is that it simultaneously sends a long-range systemic signal to the rest of the entire plant.
A warning signal.
Exactly. It is essentially screaming through the internal plumbing, warning the healthy, untouched leaves to arm their defenses because an invader has breached the outer walls.
Okay, let's unpack this. Because conceptually, knowing that a plant sends an invaluable warning signal is one thing. But actually watching that invisible signal travel in real time is an entirely different technological challenge.
A massive challenge.
How do you even begin to see something like that? Well, the researchers had to build an absolutely ingenious biological tool to capture this communication. They created a visualizer called a JISS1-LUC reporter.
The JISS1-LUC reporter, yeah.
And the way they built this is so fascinating to me. They took the promoter region of a specific plant gene called JIS1. And JISS1 acts as an early biological marker for the systemic resistance we were talking about.
Right. It's localized in the plant's endoplasmic reticulum.
Yes, which you can think of as a sprawling cellular transport network. So they took that specific plant gene and they fused it to a luciferase gene.
And for those of you listening who are wondering why the word lucifer sounds so familiar,
it is the exact same enzyme that makes fireflies glow in the dark in your backyard.
It's brilliant. What's fascinating here is the sheer level of spatial and temporal detail this glowing tool finally allowed the scientific community to capture.
Because before this paper, we knew plants communicated internally.
We did, but the actual timeline of that communication was incredibly blurry. We were essentially guessing. But with this luciferase reporter, the team could literally put the plant under a highly sensitive camera and watch the alarm travel as a wave of light.
A literal glowing wave.
Exactly. And if we walk through the exact timeline they documented, it paints a very dramatic picture. When the leaf is initially infected with the Pseudomonas bacteria, the very first thing that happens is that the local infected area starts losing its photosynthetic efficiency.
Right.
Its chlorophyll fluorescence begins to drop.
So the plant's engine starts sputtering almost immediately.
The machinery is definitely breaking down. And as that local stress builds, the leaf starts generating faint light called biophotons as its cellular lipids begin to degrade.
Okay, so it's under stress.
But the real magic happens right at about the 3-hour mark post-infection. Something incredible triggers. A glowing signal, driven by that firefly enzyme, shoots down the petiole.
And the petiole is the little structural stem that attaches the infected leaf to the main body of the plant, right?
Exactly. It shoots right down that stem.
And once it hits that main stem, it moves remarkably fast. Looking at the study's timeline, by the 4 to 4.5 hour mark, the adjacent, completely healthy leaves are glowing brightly on the camera.
They're lit up.
They have received the signal, they're fully warned, and their immune defenses are actively arming. The real kicker for me when reading this research is that all of this complex systemic warning happens before the originally infected leaf even shows any visible signs of collapsing or dying.
That's the wild part.
If you were just looking at the plant with your naked eye, the visible tissue collapse doesn't happen until about the 6-hour mark.
Long after the warning has been sent.
It really makes you think about our own bodies. Imagine you get a tiny paper cut on your finger. Before you can even visually see the redness or the scab begin to form, within just a few hours, your entire body has preemptively mounted a massive systemic defense.
Yeah.
What these researchers did is invent the camera technology to watch that biological alert light up and travel to the veins of a plant in real time.
The warning entirely outpaces the physical damage, which is the hallmark of a highly optimized evolutionary survival mechanism.
Absolutely.
But creating that visualization tool, as brilliant as it was really just the prerequisite for the true mystery the researchers set out to solve.
Right, the real question.
They had the camera, they could see the alarm system going off. The next logical question was figuring out what chemical messenger was actually pushing the button to trigger that alarm in the first place.
Who was initiating the broadcast?
Exactly. And the answer to that question brings us to a major turning point in our understanding of plant pathology.
Here's where it gets really interesting. Because historically, the scientific community felt pretty confident they already knew who the messenger was. This was essentially textbook knowledge.
Oh, absolutely.
For decades, the consensus was that a chemical called salicylic acid,
which is basically the plant version of aspirin?
right, plant aspirin. Salicylic acid, along with a few other molecules like picoic acid, were the primary drivers of this systemic acquired resistance. They were the established heroes of the story.
That was the dogma.
But science is entirely about testing assumptions. So, the researchers took their glowing firefly gene tool and engineered it into classic mutant plants.
The lab staples.
Yeah, these were plants that had been specifically bred in laboratories to lack the ability to respond to salicylic acid.
Yeah.
We are talking about well-known mutants with laboratory names like SID2 or NPR1.
Right.
These plants are fundamentally compromised when it comes to classic systemic immunity. The logical expectation was that if you put the alarm system into these mutant plants, the system would be broken.
The salicylic acid pathway was dead?
So the glow shouldn't happen.
The expectation was a dark screen, but when they infected those mutant plants with the bacteria, the glowing JISF alarms still went off perfectly. The traditional molecules failed to trigger the reporter in other tests, and stripping the plant's ability to use those traditional molecules didn't stop the reporter from firing during a live infection.
Wow.
That realization meant the real initiator, the true first responder pushing the button, was something else entirely.
And the shocking discovery they unveiled is that the true messenger is a hormone called jasmonate, or JA.
Jasmonate. And to understand why this scent ripples through the biology world, we have to look at how we traditionally categorize plant defenses.
Who do it?
Jasmonates are a class of hormones that are almost exclusively associated with defending against macroscopic threats.
You mean like big threats?
We were talking about chewing insects, herbivores, caterpillars, things that take physical, mechanical bites out of the plant's tissue.
Right.
Salicylic acid was supposed to be the dedicated defense department for microscopic microbial pathogens, like our Pseudomonas bacteria.
It almost feels like a biological case of mistaken identity. The plant is actively utilizing the bug defense hormone to fight off a microscopic germ.
It does feel like that.
And the researchers didn't just stumble into this and guess. The level of rigorous, multi-layered proof in this paper is staggering. They attacked this hypothesis from every conceivable angle.
They really covered their bases.
First, they went for the most direct route. They just applied jasmonate directly to the leaves. They also use a highly active synthetic mimic of jasmonate called coronitine.
Coronitine, yeah.
When they applied either of these directly to the local leaf, the entire leaf lit up. The luciferase reporter was overwhelmingly activated.
It was undeniable.
But As any good scientist knows, showing that a chemical can cause a reaction isn't enough. You have to prove that the plant needs that chemical to survive. You have to break the system on purpose.
You have to prove absolute necessity, not just correlation. So the team utilized another set of highly specific mutant plants to verify this.
More mutants.
Yeah. They used a mutant called a Kargios, which has a genetic flaw that completely prevents it from manufacturing jasmonate. And they used another mutant called a UKY-116, which has plenty of jasmonate, but is physically unable to perceive it.
So the receptors are blind.
Exactly. The receptors are blind. When they infected these jasmonate-compromised plants with the bacteria, the glowing systemic signal completely vanished.
Nothing.
The camera showed nothing, and the biological result was devastating. The plant's systemic immunity completely failed. It could not protect its distant, healthy leaves from the spreading infection.
That's incredible.
And to make the findings absolutely bulletproof, they stepped away from genetics and backed it up chemically.
How did they do that?
They applied targeted chemical inhibitors. Compounds with names like phenidone and gerin-1, which chemically block a normal plant from synthesizing jasmonate. Once again, the alarm failed to sound.
The glow stops, the communication goes dark, and the entire plant becomes vulnerable to the invader.
Exactly. When you look at all of that data together, it proves a critical new reality for plant biology. An early, extremely rapid pulse of jasmonate is absolutely crucial to initiate the defense against a bacterial invader.
It changes everything.
This revelation completely blurs the rigid textbook lines we used to draw between how a plant handles a macroscopic threat, like a caterpillar taking a bite, versus a microscopic threat, like a bacteria slipping into a pore.
The plant is far more resourceful than we gave it credit for.
It is using an early jasmonate burst as a universal starting gun for its systemic defense network.
A universal starting gun. That is a brilliant way to frame it. But that naturally leads us to the next massive mechanical mystery of this paper.
Oh, yeah, the travel.
Right. We know the first responder is the jasmonate hormone. But how does that chemical signal travel so fast through the plant's architecture? It's not like a fern has a pumping heart or a pressurized bloodstream moving fluids around at high speeds.
Definitely no heart in a fern.
When the researchers zoomed in on the microscopic mechanics, they found that the signal moves in a way they describe as symplastically.
Symplastically.
That means the signal is moving directly from cell to cell, passing right through the interconnected walls. And it travels specifically through the endoplasmic reticulum network within the plant's vascular system, its veins, and its outer epidermal cells.
Right.
It's essentially using the plant's internal structural plumbing as a high-speed fiber optic cable.
If we connect this to the bigger picture, this is where the research leaps from chemistry into the realm of biophysics.
Biophysics,
Yeah, because the researchers didn't just find a chemical hormone moving through the plumbing, they discovered an electrical signal.
Electricity in a plant.
Yes. Using highly sensitive laboratory equipment, they attached tiny microscopic electrodes directly to the surface of the living leaves.
Wow.
They placed one on the infected leaf and others on the healthy leaves farther away along the stem. And what they captured was an incredible electrical event.
What did they see?
Following the bacterial infection, the localized leaf undergoes a massive electrical depolarization. The amplitude drops by about negative 100 millivolts.
A drop of negative 100 millivolts. For a tiny, fragile plant leaf, that is a massive electrical shock.
It really is.
It is an undeniable electrical wave rolling right through the living tissue.
It is a profound electrical signature, one that rivals the electrical activity we see in animal nervous systems, even though plants obviously don't have neurons.
That is mind-blowing.
The researchers termed this wave a SISP, which stands for Systemic Immunity Surface Potential.
SISP.
Once the infected leaf undergoes that initial huge depolarization, it eventually repolarizes and stabilizes. But that electrical wave doesn't just dissipate, it travels.
Through the plant.
It moves out of the infected zone and travels to the adjacent healthy leaves where the SISP peaks at around the 7-hour mark post-infection. And they discover that this electrical highway is not just some random byproduct of dying cells.
What is it then?
It is a highly regulated biological mechanism that relies heavily on calcium signaling.
Right. And their method for proving the calcium connection was incredibly elegant. They took a specific chemical called LALCL3, which functions as a strict calcium channel blocker.
A blocker.
It basically acts like a cork, clogging up the microscopic pores that allow calcium ions to flow into the plant cells. When they applied that blocker to the plant tissue and then introduced the bacteria, The glowing immune signal was completely abolished.
To shut it down.
The Firefly reporter went totally dark. If the plant doesn't have access to flowing calcium, there is no electrical wave and there is no systemic warning.
And the research drills down even further into the specific hardware the plant uses to generate this electricity.
Hardware.
Yeah, this electrical highway requires very specific calcium channels to function properly. They belong to a family called glutamate receptor-like proteins.
Okay.
Specifically, the study identified two variants named GLR 3.3 and GLR 3.6 as being entirely essential.
GLR 3.3 and GLR 3.6.
Yes. When the team tested mutant plants that were genetically missing these specific glutamate receptors, the results were definitive. The plants completely lost their ability to send the SISP electrical wave.
Even with the hormone present.
Exactly. Even though the jasmonate hormone was present and trying to sound the alarm, the electrical propagation failed. It is exactly like taking a pair of wire cutters and severing the main electrical line in a house.
Oh.
The switch might be flipped, but the power simply cannot travel to the lights.
So what does this all mean? If we step back from the mutants and the millivolts and look at the sheer scale of what this paper uncovered, we have to fundamentally change our mental model of what a plant actually is.
We really do.
Plants are not passive, helpless green statues sitting in a terracotta pot in your living room or rooted in your garden.
Not at all.
Upon detecting a microscopic bacterial infection, they are capable of generating a rapid, localized jasmonate hormone pulse. And that chemical pulse couples together with a sprawling, calcium-driven electrical wave, that SISP we just talked about, to instantly warn the rest of the body.
It's amazing.
They are incredibly active, highly dynamic organisms operating on chemical and electrical frequencies that we simply couldn't see until we had the right camera.
The evolutionary elegance of this entire design is truly something to marvel at. Remember earlier in our discussion when we talked about how jasmonate is usually reserved for caterpillar attacks?
Right, the bug defense.
Well, it turns out that when a plant is actively being chewed on by an insect, it generates electrical waves to warn the rest of the plant. Those are called WASPs, or wound-activated surface potentials.
ASPs instead of SEPs.
Exactly. What this paper beautifully demonstrates is that the exact same electrical pathways, those specific GLR calcium channels, and the exact same hormone, jasmonate, that a plant uses to scream, "I'm being eaten by a caterpillar," are being masterfully utilized in a slower, more sustained way to broadcast, "I'm being infected by a bacteria."
That is so efficient.
It is. The plant doesn't need to waste energy building two entirely different internal telephone networks. It uses one highly efficient, multi-purpose biological telecom system.
It just changes the message.
It simply broadcasts entirely different types of emergencies by changing the speed, the duration, and the frequency of the signal.
It is the absolute pinnacle of biological efficiency. So for you listening, the next time you are watering your houseplants or taking a walk through a botanical garden or even just brushing past a bush in your local park, I want you to remember this deep dive.
Yeah, look at them differently.
As you look at those quiet leaves, remember that you are walking through a dense, invisible grid of high-speed electrical and chemical chatter. Millions of microscopic calculations, hormone pulses, and electrical warning signals are firing off right next to you, completely silently.
You know, this raises an important question, and it's a lingering thought based on a fascinating, almost easily missed detail buried deep in the discussion section of the study.
Oh, what do they find?
The researchers noted an incredible ecological interaction. They observed that when this specific JISS1 reporter is activated, meaning when the plant is actively sounding the internal alarm about a bacterial infection, a generalist insect, specifically the Helicopa armigera caterpillar, actually modifies its feeding behavior.
Modifies it how?
The caterpillar will physically move away from those specific leaves that are broadcasting the bacterial alarm.
That is wild. The caterpillar somehow senses the internal bacterial alarm and decides it wants absolutely nothing to do with that leaf.
It just packs up and dines elsewhere.
It just leaves.
It's an incredible observation. And it leaves us pondering something profound about the natural world. If a localized bacterial infection triggers an internal electrical and jasmonate response that simultaneously arms the plant against disease, and it repels chewing caterpillars on the outside.
Could plants be using a unified cross-kingdom language of pain?
Exactly. If the exact same chemical and electrical vocabulary is understood by the invading bacteria, the plant's own systemic tissue, and an entirely different species of insect crawling on the surface, are we only just scratching the surface of a universal distress frequency?
A universal distress frequency.
One that invisibly dictates the behavior of the entire ecosystem around it.
That is an absolutely incredible thought to leave on. A universal, invisible language of pain, shaping the movements of the whole ecosystem. Thank you so much for joining us on this deep dive. We hope you enjoyed exploring the hidden electrical lines of plants with us, and we are pretty certain you will never look at a quiet houseplant the same way ever again.